Hello everyone, my name is Ricardo Netto Goulart. I'm a medical student from the Federal University of Pelotas and I'm really pleased that you have clicked my poster presentation. This work has been studied inside the Cell Modulatory Laboratory of the Federal University of Pelotas, Brazil. The aim of the study was to seek the relation between the activity of the enzyme ADA, Adenosine Deaminase, and tDCS, transcranial direct current stimulation, in rats undergoing neuropathic pain. The ADA is an important enzyme involved in purine metabolism. It converts deoxyadenosine to deoxyinosine, an important mechanism that can indicate the activation of lymphocytes, for example. But what about tDCS? tDCS is a non-invasive, painless brain stimulation treatment that uses direct electrical current to stimulate specific parts of the brain, using low-intensity current that is passed through two electrodes placed over the head, which modulates neuronal activity. I know that you may ask, why have we chosen the ADA enzyme in specific? And I know the answer to you. We have chosen this enzyme because of a series of papers that relate the development and maintenance of neuropathic pain to the purine receptors. So, what would happen on a scenario where the object is using tDCS as a treatment? Well, that was our question. To try figure out the answer, we have reproduced a sequence of methods. Our experiments were initiated with 45 male Wistar rats, randomized into 9 groups with 3 main groups and its subsections. The main groups were 1. Injured with the constriction of the right sciatic nerve, 2. Sham, with the same procedure but without the constriction of the nerve, and 3. Control. Each one divided into 3 subsections. These subsections were the A. Stimulated group with the use of transcranial current stimulation of 0.5 microampere for 20 minutes over 8 days, B. Non-stimulated group with the attachment of the electrode to the rat, but with the electrode disconnected to the battery, and C, which one had no electrode attached. After the experiments, the rats were euthanized. There was the blood collection and centrifugation for the obtainment of serum. Our way to measure these relations and to see if the results were significant was the one-way ANOVA, followed by student Newman kills. But what have we learned from it? The data shows the effectiveness of the tDCS treatment to pain. Why can I say that? Because it was able to increase the pain threshold in the rats submitted to the chronic pain model, with a p-value lower than 0.05. However, it was unable to modify ADA activity in the control and sham groups. By saying this, we know that further studies are needed to elucidate the relation between this enzyme and tDCS. The key words to this work were ADA, tDCS, and neuropathic pain. Our acknowledgements goes to Federal University of Pelotas, Cell Modulatory Laboratory, and the Bioengineering of the Clinic Hospitals of Porto Alegre. We also had the financial support from CAPES, Coordination for the Improvement of Higher Education Personnel, and CNPq, National Council for Scientific and Technological Development. It was a pleasure to demonstrate our work here. Thanks for the opportunity. If you want to contact me, you can find me on LinkedIn and on my email, that is ricardonettogoulart@gmail.com.